Okay, so good morning, everyone. So just a few things to remind everyone that the rough draft is due next week, right? So that's um, as a rough draft, and I do mean the complete paper, but obviously not going to be with the fully final figures and things like that. What I want to see is that there's a clearly developed story, that there's connectivity between the ideas. And so I will read those rough drafts and quickly then set up a meeting to meet with each team to then work on how we translate that to the actual complete, you know, more formal draft. So today, what we're going to do is continue our discussions about the use of metals and metal complexes for an anti-diabetic purpose. And so to put this into context for what we were discussing last time, where we were looking at vanadium oxidation state 4 primarily, but also vanadium oxidation 5, how that metal serves as something that can enhance insulin sensitivity. We're going to now look at it from the perspective of chromium-3, this particular chromium-3 complexes, and how they may enable an anti-diabetic behavior through a different mechanism. And so just to review what we discussed last time. So this is going to be very important, this idea of how insulin really enables signaling. So from this perspective, as we discussed, the insulin hormone will interact with its receptor. And that interaction then triggers this activation of the insulin as a kinase that can then lead to this phosphorylation series of events. That serves as a signal to ultimately the glucose transporter in order to be able to import glucose into cell and to metabolize it. And that way, we're able to control that excess of glucose or that glucose that's coming from a diet, and we'll be able to meet our demands. And so this is a perspective, or at least the working theory about how this complex, that's abbreviated BEOB, how that complex may exhibit its anti-diabetic effect via the transferrin mechanism. And so the understanding is that to some extent, the vanadium ion has to interact with the transferrin. Whether or not it retains its ligand is not necessarily clear. What is understood is that vanadium will ultimately be brought into the intracellular environment, and there it has to interact with the PTB1B cysteine, so that you essentially will block that cysteine from being able to cleave the phosphates that are part of the phosphorylation cascade. And so by inhibiting that process, you essentially will enable an increased sensitivity towards the insulin and insulin receptor. So obviously we're talking about insulin being able to dock at its receptor and allow for this process of translocation of the glucose transporter and the uptake of glucose and then subsequent metabolism. What we're going to look at today then is how people have explored chromium for its properties. So when you go to the store, let's say you go to Walgreens, you are able to find chromium sold, I think it's typically as chromium citrate or chromium picolinate, as a potential supplement. So it's not something that's FDA approved, but it is for the purposes of trying to lower your sugars. And so that is how it's advertised. And we have been introduced to chromium since the beginning of the course, where we looked at chromium in terms of its oxidation states and what impact it can have. And so it turns out that chromium-3 is the most stable form of this metal. It can exist in +3 and +6, as we are aware. There are examples of +1, +4, and +5. But focusing on chromium-3, when we're talking about the coordination chemistry, we typically are talking about octahedral complexes. And so it's believed to some extent by some people that chromium can play an important role in terms of this insulin sensitivity. And so there is a bit of a debate, I would say that no longer exists, but of whether or not chromium is an essential element. And so we'll look at that as we look at some of the material from today. So it is debated whether it's essential. I would say today is generally understood not to be an essential element. And in excess, it becomes quite toxic. So this has been the general dietary recommendations if you want to see some sort of effect, where it's stipulated for women and men, the amounts of how much you should intake depending on your age, and then how much should be the you would intake if you're a child. But in the European Food Safety Authority, they do not consider chromium an essential nutrient. And so they have long championed that it is not essential, although I think the US has basically caught up to that. And again, I'll show you some important results that has led to that understanding. All right, so this was an important study, and I'm presenting this as if, just to perspective. So in the 1950s, Schwartz and Mertz were working with the rats that were consuming this yeast-based protein, and they developed an inability to remove glucose from their bloodstream. And so in this study, they explored what different metals could potentially have an impact to help improve the processing of this glucose that was in the bloodstream of these rats. And so a number of different elements, many of these being metals, were tested. And it was observed that the administration of chromium as a salt, chromium chloride, supplied via drinking water, so this is delivered orally in this dosage here, okay, or intravenously, so at this dosage, resulted in an effect where you could decrease the amount of glucose that was present in the bloodstream. So for perspective then, chromium-3 here, it was delivered orally, or at least a portion of this was delivered orally. So looking at the aqueous speciation, pH-dependent aqueous speciation of chromium as it was delivered as a chromium salt, we can appreciate that at low pH, you essentially have chromium that is hydrated. So that's represented as just free chromium. We're aware that that wouldn't just be free-floating metal, it's be surrounded by solvent. And so then you see how that speciation changes as you increase pH, where it becomes more pronounced in terms of the hydroxide species that would exist. So it turns out that the chromium absorption would be non-saturable because it would go through a passive diffusion mechanism into the rat small intestines. But the amount that actually gets absorbed into the bloodstream is a very small percentage. We're talking about less than 1%. Okay. But that is in comparison then to chromium complexes. So some examples being the chromium complex of the nicotinate ligand and also the picolinate ligand, where that can increase it. And we're not talking about super big amount, but you increase it to 1.3% to roughly 3%. So complexation is important in terms of improving the ability to potentially utilize that metal. So we're looking at the aqueous dependent speciation here. We're talking about chromium hydroxide or dihydroxide being the predominant species that would be physiologically relevant. Okay. And so, excuse me, one last thing, although I'm going to clear that up. A very important note is that about almost 40% of chromium will hydrolyze and polymerize in the intestines, and that also contributes to why you have a relatively even lower amount of absorption into the bloodstream, just because a lot of it will just sink into this insoluble material. So people have explored chromium complexation for its potential as an anti-type 2 diabetic agent. And so I mentioned to you this complex, you can see it commercially available as a sort of supplement. And so it has been observed that it can improve glucose uptake in insulin-resistant adipocytes, so these are fat cells. It has a capacity to inhibit PTB1B, although it's not clear how it's exhibiting that particular effect. It's also capable of generating hydroxy radicals, so something similar to what we have observed with vanadium, although it's not entirely clear for chromium-3 how it's generating that, although you have all mentioned some of this features about chromium and its capability of potentially being toxic because it can induce oxidative DNA damage. So this other complex, the chromium with the phenylalanine amino acid. So here we just get a depiction of that complexation there. So that also has been observed to enhance insulin sensitivity by amplifying the phosphorylation of the insulin receptor substrates in adipocytes. So it contributes to, apparently, to that phosphorylation cascade. It's also been observed not to induce DNA damage. So presumably, that coordination will protect the oxidation state, basically preventing potential redox activity of the metal center. All right, but another thing that's understood is that chromium also has to interact with transferrin, as is also proposed for the mechanism of action of vanadium. That a significant amount of it goes through the transferrin protein in order to enter into the cellular environment. So here we're just looking at the binding constants associated with chromium association with this protein. It's proposed that this metal coordinates to the protein in a manner that's comparable to iron. What I mean by that is you get this very similar coordination chemistry, but the affinity constants are lower, significantly lower. Okay, if you remember, for iron, both sites were above 10 to the 20. So we're talking about many orders of magnitude different. And in fact, at below pH of 6, the C lobe of this protein can readily release that chromium-3. And so why I mentioned that below pH 6, if you recall the endocytotic process of how transferrin brings in iron into our cell, it's important that they have a reduction of pH plus a reduction of the metal for release. Well, reduction of pH is enough to just kick off the metal. You don't really need to do anything about the readout of the oxidation state of that metal. Although it's not entirely clear how, once it's released, how the metal actually can escape the endosome to finally enter this intracellular environment. That's something that people still are not entirely clear about. But it's believed that the function of chromium in terms of how it can potentially influence insulin sensitivity is related to its interaction with a protein called chromoglobin. So here we're getting a perspective of how this interaction might play. So in the 1980s, Wada et al., they discovered a unique chromium-binding protein called chromoglobin. As far as I understand, there's still no crystal structure of this protein. It's 1,500 Dalton. It is observed to bind as many as 4 equivalents of chromium-3. And so what's understood is that the process of chromium interacting with transferrin seems to be influenced by insulin, possibly because of this association with the chromoglobin protein. So we have this sort of visual of insulin-sensitive cells and how that can influence the translocation of the endosome that contains the transferrin receptor so that it can go on to the to reach the plasma membrane. So you have plasma membrane recycling of the transferrin receptor that is sensitive to insulin, again, for insulin-sensitive cells. All right, so here's a perspective to give us this idea of what potentially that chromium chromoglobin interaction is doing. So here we're looking at this chromium, some source of chromium that will deliver the chromium to transferrin. So now you have this transferrin complexation with the chromium. And then you have an interaction with the transferrin receptor that triggers in the endocytotic uptake of the chromium. And so now you have this in this endosome, and then you have the what I mentioned to you, the lowering of the pH, which is sufficient to remove the chromium. There may be some other component like chelation that also helps the chromium to be released, and that leads to the chromium available for chromoglobin. There goes that breathing again. All right. So what we're looking at here then is how this may or may not influence insulin. So this is another model, for what is appears to be an important feature of that chromoglobin, activation by binding to chromium. So what we're seeing here is we already have the chromium now in the cellular environment. It forms into the hollow protein. Hollow protein means metal bound. And then you have this insulin that is already bound to the insulin receptor, and a possible association of chromoglobin with the insulin receptor to influence that activation of the actual insulin receptor to then bring in glucose. So that is a module that's proposed to how chromium may be influencing that sensitivity towards insulin. And so here's just another depiction of that very same process, trying to bring this all together, where we have the idea of chromium going into this cell environment, and ultimately how that might modulate this interaction so that ultimately you're able to bring in glucose through the glucose transporter or a glucose transporter and metabolize that molecule. All right, so I'm going to focus on a few papers because the literature on chromium is not as, you won't necessarily find it in text, so you'll see mentions of it, not like you would find for vanadium. Vanadium was more classically studied. Chromium, more you will see it in the actual literature. So an important person in this field, go ahead. Yes.
There's some possibility of chromium coordinating to transferring or something?
Well, it does coordinate to transferrin. So it does coordinate to the metal binding site. So that's it's understood to be a mechanism, may not be the only mechanism, but a mechanism by which it can enter into cells.
Yeah. Well, I was thinking when you said that, once like the coordinates from the
Let's go back.
Yeah, because you mentioned that at some point it's like there's free chromium and it's no longer how it leaves the cell.
Right. So that's the part that's not clear. So even all these figures, that part is just like it comes out of the endosome. So it could be some sort of ligand. This is what's also proposed for non-redox active metals, that there are chelators that can literally cross the endosome. It can pull that metal, and possibly because of the receptor recognizing the ligand itself, that you can basically pull the metal through. That's an understanding of what might happen.
Okay.
Especially if we're talking about a pH of 5.5, right? That's relevant to the endosome. So what I was mentioning was that Professor John Benson, he's from the University of Alabama. So he has been one of the key people in the United States to really push this idea that chromium is something that's not essential. And so this was a very seminal experiment or study that he did with his team, obviously, to kind of show that chromium, in terms of the properties that exhibits, appears to be more of a therapeutic nature than an actual something that is an innate function of it, in terms of how it interacts with biomolecules. So this is a study, chromium is non-essential trace element for mammals, effect of a low chromium diet. All right, so I'm going to contextualize it with results that are prior to this actual study. So Anderson and co-workers tried to establish that chromium-3 is an essential element by evaluating a series of rats. So these were Wistar rats that were fed, and according to their experiment, chromium deficient diet. Because that's typically how you establish if something is essential, is you basically try to remove it from diet, and then you try to see what effects physiologically it has on the body, and then by supplementing back in, does that reverse the effects? So something similar to that was designed here. So rats were placed in a plastic cage environment with no access to metal components. The diet consisted of 55% sucrose, 15% lard, 25% vitamins, minerals, and then 33 kilograms per kilogram diet that contained that chromium. So the high sucrose was to produce a carbohydrate stress to further induce a chromium deficiency, as it would lead to urine excretion. That was the idea of that excess carbohydrates. So the administered low copper concentrations for the first 6 weeks to compromise pancreatic function, and then high iron concentrations throughout to further aid in chromium deficiency. So then you had in the study a pool of rats that were supplemented with water that contained 5 ppm of chromium chloride. All right, so this was the context. Basically, create a situation where you would, in theory, induce a chromium deficiency. So then an intravenous glucose tolerance test. So a glucose tolerance test is basically that you're going to subject an organism, an animal or humans, to just a sudden amount of glucose concentration into the bloodstream. And that's typically done when you fast the animal or you fast a person. So it has been on a diet for 24 weeks, the diet referring to this chromium deficient diet. So the plasma insulin levels were found to be higher in chromium deficient rats. So the various stresses of the diet may have caused hyperinsulinemia, which the chromium supplementation could revert. So here, this was the observation, was that basically by now incorporating chromium, you can reverse that effect of the insulin itself. However, and this is where John Vinson's work comes into play, they looked really closely at the experimental design of this work because there was a few flaws in this work. And the flaws was in the thinking that the actual experimental setup was actually chromium deficient. So they did this by just sort of a theoretical calculation. So here, humans need a daily intake of 30 micrograms of chromium. That's just the proposed need, right? If it is something that is essential. So if we calculate this for a 65 kilogram person, this would amount to approximately this amount of chromium requirement per kilogram body mass per day. So if you look at the study and how they design it, we had a 100-gram rat that was eating 15 grams of food containing 33 micrograms of chromium per kilograms of food, which would amount to 5 micrograms of chromium per kilogram of body mass. So we're talking about easily an order of magnitude higher than what would be even something that would be considered enough for humans. So what this would suggest, right, that if this math is correct, that this diet was never chromium deficient at any point. It was always enough chromium there to have had an effect. And so if there was an effect that was observed, their rationale that this was because the supplemented chromium was having a pharmacological effect, so in the therapeutic effect, not because you were adding the, compensating for any deficiency of chromium. All right, so a study was done to try to verify this. So basically, think the same type of approach as before, but trying to do a much more effective job of minimizing the amount of chromium. So design conditions with as low of a chromium content as feasibly possible. So the key thing that was that these conditions could not sacrifice the content of other essential things in the process of trying to achieve a low chromium content. So that could be quite difficult. So what was done here? So 32 sucker lean rats were used that was 6 weeks of age. They were, so they had different pools of rats that were used. Some that were treated with they were all treated with a purified diet, but some that would just have the purified diet, some that had the purified diet with chromium sufficient diet. So it was supplemented in the form of chromium sulfate. So that salt of chromium is a chromium 3 salt. And then rats purified with chromium sufficient diet with an additional 200 micrograms of chromium per kilogram. Then you had another pool of rats that had an even higher supplementation, which was 1,000 micrograms kilograms per, keep saying that, micrograms chromium per kilogram. Another thing that was important in this experiment was the housing. And the housing was to try to keep it as minimally metal containing as possible, but still hospitable for the rats, right? Because you want them to be comfortable, reasonably speaking. And then what data was collected? Well, the weight of the rats, because that's always a signal that something's going wrong. If their weight changes, either up or down, that's an indication that something's not necessarily right. We also observed the food consumption, recorded every 2 weeks, so twice a week, and then the intravenous glucose and insulin challenge at 23 and 25 weeks after fasting between 10 and 12 hours. So those are the parameters that are being measured. All right. So this is an example of the housing, right? Try to keep it as minimal chromium containing as possible. So you put in this plastic container that is breathable, right? Because you don't want them to suffocate. Your experiment would fail. That material at the bottom that's comfortable, but that has a very low amount of chromium because most likely they might actually chew on it, right? And then the source of, I'm assuming that's where it was supplied, the water. I'm assuming that I can't remember, but I don't think it necessarily had a metal connector, and then the food being supplied to them. All right, so the parameters. So we're looking at body mass here, and we're looking at it over time, and then the different conditions, right? No chromium, chromium sufficient, then the chromium sufficient plus the excess chromium. And what do we see, Brenda? What are we seeing here? Increase the body mass and no dependency on those conditions. They're basically the same with an error. Okay, so body mass is not getting affected. Can you see that too, Oscar? Okay. All right. Then the next thing they did was to do a glucose tolerance test. So again, a glucose tolerance test, you do it after you fast. Fast means that you don't feed them, and they said 10 to 12 hours. You inject a dosage of glucose, and you see how the body responds. So basically, you're tracking that glucose uptake and then the use of that glucose over time. So here we're looking at the glucose level, time after injection. So time 0, this is where the basal level of glucose is at, and then obviously we're looking at the different conditions here, and then we're tracking in 20 minutes. You have a spike. Javier, why is there a spike? Well, it's a spike because you injected the glucose. So it's going to take some time to register, right? Because it's going through the bloodstream. So 20 minutes, you see going from 100 milligrams per deciliter to then about 3 times. So you get that spike within 20 minutes. And then over 120 minutes, you start to see it being consumed, right? So then the way they calculate, because you have to calculate the amount of glucose in this time frame, what they do is basically take an area under the curve. So they measure all this to calculate area in the curve, and that converts to this information over here, which is the amount of glucose under the curve under the different conditions, okay? And in terms of statistics, they're basically not very different. Maybe that chromium sufficient was a little bit higher, but within error, they're all basically the same. So this did not have an impact in terms of the glucose.
luego de que llega al punto máximo de la inyección, él se va degradando o desapareciendo.
Well, not degrading. So imagine que you have your insulin is going to be released. So it's insulin is now going to then signal que tenemos glucose sangre. So you're going to bring that in. And so that's going to burn inside the cell. So the more, as it gets burned in the cell, metabolized, the amount that's in the blood, because this is glucose in the blood, is going to be lower. Yeah, so degrading, but not degrading in the sense that it's degrading in the blood. It's actually getting used by the body. In this case, these rats. Okay. Another experiment was an insulin tolerance test. So an insulin tolerance test, like the glucose, is you fast the animal 10 to 12 hours, and then you give them a shot of insulin. Okay, and then you see how does the animal respond. And so here we're looking at it from the perspective of glucose. Okay, so insulin was injected, but we're seeing how the glucose basal level is getting affected. All right, so let's do a little comparison. So remember over here at time 0 is around 100. So at time 0, we're still around that same level. Okay, so everything seems to be checking out. So we're looking at the glucose level, and you shot in insulin. So within 20 minutes, you see this drop, right? And then because of the insulin, and then you see that that glucose starts to decrease over time. All right, and then you see the different conditions again in terms of the amount of chromium. So taking the area under the curve again, area under the curve, we see that we don't have a difference in terms of the chromium and how much glucose is available in the blood collectively over time. All right, so there's no observed difference in terms of total plasma glucose. Let's look at it from this perspective then. Now we're going back to the glucose tolerance test, and what we're tracking here is not glucose, we're tracking the insulin, okay? And so here, you have your glucose tolerance, so you give them a dosage of glucose, all of them, and you're seeing how the insulin of the actual rats, so this is the insulin inside them, is getting affected. And so what are we seeing is there does appear to be some amount of effect depending on chromium. So doing the area in the curve, we look at the insulin levels, and what do we observe here? So what we observe is a higher amount of insulin in the situation where the diet with no chromium, and then you have the sufficient chromium, there's a decrease, but there is a pronounced decrease with the even higher amounts of chromium. So there's a dose response of a decrease that's associated with insulin. So this suggests that the chromium is impacting the insulin sensitivity. Go ahead.
With the other ones, it really doesn't affect the glucose levels. So like the glucose levels are going to be related to the insulin, so there's not a meaningful effect.
Right, because there's going to be like a sort of a cap of how much insulin will really affect how much glucose is getting metabolized, right? Because there's only so much insulin receptors, insulin interaction. So this is basically telling us the effect it's having, but that in itself won't necessarily affect how much glucose overall is going to get consumed. It will just show us that we basically have a normal performance in terms of the glucose being utilized. So yeah, supernutritional levels of chromium increases insulin sensitivity to suggest a pharmacological effect. That was what their conclusion was. All right, so what I'm going to show you is a couple of papers that try to probe a little about mechanism of action and just overall effects that chromium complexes can have. So here we're looking at the anti-diabetic properties of chromium citrate in alloxin-induced diabetic rats. So alloxin is something that can do something very similar to the STZ compound that I discussed last time in terms of affecting the pancreatic cells. So here, Lee et al. Decided to work with citric acid as a ligand for chromium because citric acid is something that is biorelevant and is believed to be the type of small molecules that can coordinate with metals in blood, but also within the cellular environment. So that's what's kind of the thinking here. So what they wanted to do, in addition to structurally characterizing coordination chemistry, was also to understand the potential effect that the chromium citrate could have in terms of insulin sensitivity, and also seeing any toxicity associated with the chromium complex. So in the paper, they refer to chromium citrate as a chromium 1 citrate complex. So what I'm showing you here is not what they're proposing. What I'm just showing you here is the coordination chemistry of citrate with this metal center, where it's coordinating through, and this is very characteristic of the citrate molecule, through two of the carboxylates, so the beta carboxylate and through that beta hydroxide. So those are the characteristic coordination sites to chromium, and that's true for iron and for many different metals. But there are a few metals like titanium and other hydrolysis prone or more hydrolysis prone one that the coordination switches from being a tridentate ligand to a bidentate ligand. Predominantly, we're talking about interactions with the beta carboxylate and also hydroxyl ligand. So that's why I wanted to show you this figure, not because that's the actual compound that they are proposing. All right, so they worked with these rats, and these are approximately about average weight of 20 grams. These were the experimental conditions, temperature, let's say that that's room temperature, average humidity. They were allowed free access to food using standard pellet diet and distilled water. So these were the different rat groups that were examined. So they used alloxin as the molecule that would induce the diabetic model. So this is just basically the intraperitoneal. So intraperitoneal is like right here, just to the gut, injection of that alloxin, and that induces the diabetic state. So then from there, they have these different groups. One is the control group that did not get this alloxin treatment. So they don't have this diabetic condition. Then you have the alloxin treatment. Then you have a positive control, which is you induce the diabetic model, but then you inject this molecule, this glibin-clamide molecule, which aids in insulin production. And then you have chromium chloride and do a certain dosage of it, and then chromium citrate at different dosages of it. The only numbers I'm reporting here is the effective chromium content, not the chromium citrate content that's actually being delivered. All right. So again, what are the parameters that are being studied? Looking at body weight, blood glucose, and they looked at it from 0 to 21 days. And they also looked at things with regards to the blood, the triglyceride levels, and also the total cholesterol levels. And they also looked at the amount of glycogen formed. And remember, glycogen is the polymeric form of glucose, and that's basically an indicator that your body is metabolizing the glucose and also storing it effectively. All right, so they collected blood, and they were examining the amount of glucose in this. So what we're looking at here is the blood glucose at basically time 0 and at 3 weeks later, so 21 days later. And so what we're seeing is in the non-alloxin rats, so the ones that are not diabetic, they have this normal glycemia, right, about 5 millimolar. And then what we're looking at is the amount of glucose in all the rats that do have the alloxin induction. And you basically see that the very high elevation of blood glucose levels, so 20 millimolar there. Then after 3 weeks, that's where we start to see the effect. We wouldn't see it immediately. What do we observe? So obviously the control still at the normal glycemia. The control with just the alloxin, very high level of glucose, and then you see to some extent a dose response of in decrease of glucose due to the concentration of chromium citrate. It isn't particularly pronounced and not exactly very different from the chromium chloride, but you can see that impact, and you also see the effect relative to that molecule that can induce insulin production there. So that one seems to be the most effect of them all. But within error, they're about the same. Okay, so then here we're looking at the triglyceride levels, and so we're looking at just at the 3 weeks later. So at the time of seeing the full story. And so what do we see here? So we see the amount of triglycerides that's normal. Okay, and we see compared to the level of the alloxin control. And then what we observe is that a decrease in the level of the triglycerides that appears to be dose dependent. And then you have the control, that molecule that helps to stimulate insulin production. You see that it's going even a little bit lower than the triglyceride levels in the non-alloxin rats. So there is a correlation between chromium supplementation and what effect it's having on the triglyceride levels. And then something similar is observed here for the total cholesterol levels, where we see that the total cholesterol levels basically become close to the level of the of the normal rats, the ones that are not diabetic. So there is some amount of impact that the chromium is having, not too pronounced the dose response. It basically is the same with an error. And then finally, we're looking at the total glycogen levels. Again, glycogen is an indicator of the storage of the glucose. And so in the normal rats, we have a high amount of glycogen, okay? Then compared to the diabetic rats, like about half the amount of glycogen in the liver. And then you can see here the effect that the chromium is having as it is increasing the glycogen level. It's not at the level of normal, but it's definitely higher than the untreated rats. And then compared to then the that molecule that stimulates insulin production. So the chromium citrate definitely has a greater impact in terms of inducing the glycogen levels. Okay, so the last part of this was just assessing the potential toxicity of the complex. So one thing I mentioned to you before is that chromium-3 has the capacity to facilitate oxidative stress, so that's what they were assessing here. So they wanted to look at it from the perspective of DNA. By potentially inducing oxidative stress, is there going to be any DNA damage? And so here basically is the readout of the acid that they use. So they're looking at it here from the perspective of the Fenton chemistry, because Fenton chemistry, if that's something that you stimulate, you can lead to the formation of reactive oxygen species like the hydroxyl radical. And then what they're basically looking at is treating the with DNA under acidic conditions and heat, and then seeing if there's a production of this product, this MDA product, aldehyde. And so that's an indication of whether or not there's an actual oxidative damage occurring with DNA. And they also did this in the presence of ascorbate and hydrogen peroxide. All right, so picture the same protocol, but now being done with chromium citrate, DNA, ascorbic acid, and hydrogen peroxide. If it's not clear if the chromium itself is what's going to stimulate the activity, but nonetheless, that's what they're assessing here. So the same protocol where they're trying to track the formation of this chromogen, which is an indicator of this, that the DNA may potentially be damaged because you see the formation of this product here. And so what they're looking at is what happens when you have the chromium citrate present at a dosage that's comparable to the other experiments that they're doing versus ferric EDTA, which is a positive control because it is redox active. And so what they observe is that the chromium citrate treatment versus the, just control here, you get basically something that's not above the background. But in the presence of the positive control, you really see this elevated amount of chromogen formation, which suggests that the chromium citrate is not inducing the oxidative stress to damage DNA. That's what this assay is telling us. But another way to assess this, because this is just basically an assay that's in vitro that can be done, is to look at the actual rats and see, looking at them in terms of a histopathological experiment and see what happened at the level of the tissue. So what they did is same experiment, but treating the rats with chromium citrate at a dosage that's comparable to the experiments they were doing previously. And they're assessing at the tissue what's happening. So what we're looking at here is the profile of the actual. So what are we're looking at? We're looking at the liver. So these are liver, and these are kidney. The left is the not treated animal, and then the right is the treatment with chromium citrate. And then what they're saying is that there's no actual histopathological damage being done, as it looks very similar. There's no structures being damaged in the tissue, suggests that there's no toxicity associated with this oral dosage of the chromium citrate. Okay, so essentially, this goes to show that the complexation with citrate, you can induce something that's very similar to what I had mentioned about chromium phenylalanine, something that is capable of inducing an insulin sensitivity without compromising the, not leading to a toxicity associated with oxidative stress. But this other study here tries to get us a little bit more close to the mechanism of action. So what we saw before is just this physiological effect. But here we're trying to understand a little bit more about what exactly is the chromium doing. So this is titled Chromium Enhances Insulin Responsiveness via a kinase called AMPK. All right, let's look at this more closely. So what they wanted to look at was whether chromium-3 protected against physiological hyperinsulinemia, I'm not sure, I'm struggling with that word, induced plasma membrane cholesterol accumulation. And so they also want to look at whether it protects against the cortical filament actin loss or F-actin loss, and focusing on a muscle cell line. And then they want to look at if it also protected against insulin resistance in the L6 skeletal muscle microtubes. So these have an overexpression of the glucose transporter 4. So these are the main things that they wanted to assess and to try to see what exactly is the chromium doing. So what they did was they investigated whether AMP kinase activity mediated the beneficial effects of chromium-3 picolinase. So they went focusing on this complex. So for perspective, this particular kinase phosphorylates and inhibits 3-hydroxyl-3-methylglutarol coenzyme A reductase, or abbreviated HMGR, which is the rate-limiting enzyme in cholesterol synthesis. Okay. So they're looking at this because there's an association between cholesterol affecting sensitivity. So that's what they're trying to look at it in a bigger picture. All right, so the breakdown of the actual experiment that they did. So these were the skeletal muscle cells that they were working with that had that overexpression of the glucose-4 transporter. So I'm going to just guide you through this so that you can understand, what this is really saying to us. So there's going to be one line of cells that are not treated with any chromium. So that's why there's an X here. They're not treated with any insulin, even at a long amount of time or even a short amount of time. So that's one line of cells that they're going to work with. Then you're going to have cells that are not treated with chromium, not treated with a long time frame of insulin, but are subjected to this quick dosage of high insulin over a short amount of time. And then you're going to have those that are subjected to chromium, insulin treatment, with and without that short high dosage. All right, so that's basically what we're looking at here. And now just draw our attention to this figure over here and looking at what's happening. So what are we focusing on over here? We're looking at the glucose transport. So that's basically the measure of the glucose levels and whether or not they have that insulin shot. And so we're looking at then control versus the ones that have had that 12-hour insulin with and without chromium picolinase. So it gets a little bit, you have to kind of track what exactly are we looking at here. All right, so that plus minus at the bottom is the 20-minute 200 nanomolar insulin shot there. All right, so let's assess what's happening. So focusing on over here, okay? So these cells, no chromium and no 5 nanomolar insulin over 12 hours. When you put that 100 nanomolar insulin for 20 minutes, you see that there is an increase in the glucose transport. So that's an indicator that there's an uptake of the glucose cell. But then the cells that actually have been treated with the 5 nanomolar insulin for 12 hours, okay, so they actually have gone to that second step here, with and without the 20-minute of the 100 nanomolar, you see that the levels here versus here are actually lower. So there's a lower amount of glucose transport, right? So there appears to be some sort of defect. And then to the right, we're looking at what happens when you have chromium picolinate available and what response is there. So again, the animals, excuse me, I don't know if I said animals, the cells that have not been treated with insulin and then have received, so have not been treated with insulin, but then whether or not they receive that final insulin shot in the presence of chromium picolinate. And what we see is that here in the presence of chromium picolinate, you have the same level of glucose transport. So that it does appear that the chromium picolinate is influencing the transport of the glucose here. But then we look at all the way to the right, where you've had the additional 5 nanomolar over 12 hours of insulin, you see that you are able to retain that same level of glucose transport, which suggests the following, that the chromium picolinate is helping to repair a dysfunction that's associated with insulin. Okay, so what exactly is that insulin dysfunction? It's what they believe to be a hyperinsulinemia induced effect on the glucose transport. All right. So again, the same type of experiment here, but now we're looking at not the glucose itself, we're looking at the transporter. the glucose transporter 4. And so we're seeing what happens in the presence and absence of the chromium picolinate. And so I'm going to just go this a little bit faster here, where what we observe is that the glucose transporter 4 translocation is negatively impacted by the presence of more insulin. We have less amount of translocation relative to without the that extra amount of insulin. But how the presence of chromium picolinate is actually able to increase the amount of the glucose transporter. So it induces a repairment and appears to be a repair that has to do with the insulin itself. All right, so here what they were looking at is examining chromium picolinate in terms of just other properties, the effect on the membrane and cytoskeletal defects. So here they're focusing on the levels of the cholesterol and also the levels of the F-actin protein. F-actin protein has to do with the structure of these cells. And so we're looking at what happens in the presence of chromium picolinate in the presence and absence of 12 hours of the insulin treatment. All right, so let's draw our attention first to this part here without the chromium picolinate in terms of the cholesterol levels. So we see that because of the hyperinsulinemia, we have an actual increased amount of cholesterol, right? So there's an association with that that's leading to more cholesterol being produced, which when you do dose with chromium picolinate, you basically get the normal levels of the cholesterol. So the chromium picolinate is influencing the amount of cholesterol that's being developed. Then you see something on the level of the F-actin, where here we're looking at the presence of the insulin, and that insulin defect is resulting in a decrease in the amount of the F-actin, which the chromium picolinate appears to be able to rescue that defect, right? So this is showing us what potential impacts the chromium might be having, right? So let's look at a few things here. So the hyperinsulinemia induces a cholesterol-laden plasma membrane that causes F-actin dysregulation and insulin resistance. Studies have reported that chromium-3 treatment increases AMPK activity, AMPK enzyme that plays an important role in cellular energy homeostasis, and inhibits cholesterol synthesis and improves insulin sensitivity and protects against actin loss. And so that's the data that is in line with what has been observed. All right, so here they're trying to examine that the impact that chromium picolinate has on insulin signal transduction. So we're looking at whether or not chromium picolinate can amplify insulin signaling. And so the way they're assessing that is by the phosphorylation of proteins. So remember, the insulin, when it interacts with this receptor, triggers phosphorylation cascade. So we essentially are looking at that. We're looking at a protein. So the identity of protein is not as important as the fact that it's getting phosphorylated. So it's AKT protein and then the phosphorylated version of it. And so we're seeing what's happening in the presence and absence of chromium picolinate. And so what's observed is that you basically have no difference in terms of the levels of this phosphorylated protein in the presence of absence of chromium picolinate, which suggests that insulin signaling, the signaling itself, was neither impaired by the hyperinsulinemic conditions nor enhanced by chromium picolinate. So the chromium picolinate is not affecting the signaling itself. And that's also observed in this other study with another protein that also gets phosphorylated, where the levels of this, and we're doing this by Western blot, is also not getting affected. And so that's their assessment there. But then they're trying to examine the effect that chromium picolinate has on AMPK activity in general. All right, so we have control and hyperinsulinemic conditions, which showed a chromium picolinate stimulated increase in the activity of this kinase, as emphasized by increased phosphorylation of the catalytic alpha subunit at threonine 172. So they're claiming that we're looking at activation of this enzyme, and activation of the enzyme is because of phosphorylation of that threonine. And so they say that this is an indicator, because they have a much more, a darker band, is an indicator that you have more active enzyme due to the presence of chromium picolinate. So it's a little bit subtle, but according to the measurements, that's how much activated kinase. So there does appear to be a correlation between the presence of chromium picolinate and the activity of the actual kinase itself. So further along the lines of that, the phosphorylation of the acetyl-CoA carboxylase at serine residue 79, which is a substrate of AMPK. So this was evaluated, again, to see the impact that chromium picolinate is having. And what's observed, again, is a little bit subtle. We're looking at, in the presence of chromium picolinate, that there is, in fact, a 17% increase in phosphorylation of that protein. So that protein is the substrate of AMPK. And so that suggests that the chromium picolinate is influencing the activity of this enzyme, and therefore one of its substrates is converting to its phosphorylated form. All right. So another thing they want to examine is, well, what happens if you knock down the kinase? So what that means is you basically lower the amount of active kinase available. So they evaluated the requirement of AMP activation for chromium picolinate action by doing an siRNA-mediated knockdown of the catalytic alpha subunits of AMPK. And so what we're looking at here is the levels of that particular unit of the AMPK. So we're looking at the levels of the alpha unit, that is the catalytic unit. And so from the use of this knockdown method, you can see that there's effectively less of this catalytic unit. And so they observed a substantial decrease in stimulatory effect of chromium picolinate and of an LCAR, which is a positive control on AMPK, threonine 172 phosphorylation. So along the same lines, then, to assess, you knock down this active enzyme, what effect is it having? So they were looking at here, so I kind of jumped the gun a little bit. I meant to read this here. They were looking at this experiment here, where they're focusing on the phosphorylated AMPK versus AMPK, which is not active. So phosphorylated is active. And they're assessing what happens in the presence of the chromium picolinate. And what they're observing is that because of the knockdown of the enzyme, the chromium picolinate is really not having as much an effect on the activation of this actual enzyme, because you have a knockdown of it that's been done. All right, so here they're looking at the effect of AMP knockdown on chromium. Okay, so again, focusing on the reduction in AMPK alpha did not impair basal and insulin-stimulated insulin receptor signaling. So again, it's not the signaling that's getting affected. They also observed that AMPK activity is required for the protective effects of chromium picolinate against this hyperinsulinemia-induced glucose transporter 4 and glucose transport dysfunction. So essentially what we're looking at here is the effect of the knockdown, and then what's happening at the level of that, one of those proteins that becomes phosphorylated. And so you're seeing that you have a knockdown, but still you have basically the same amount of the phosphorylated protein. So again, the emphasis being that this impact of chromium picolinate has nothing to do with the insulin signaling capability. It has to do with the impact that it has on the activity of the AMPK enzyme itself. Okay, so collectively, what they were able to determine was that the AMPK activation is an important mechanism of chromium-3 action in insulin-resistant skeletal muscle cells, and that membrane cholesterol influences cytoskeletal structure, which is essential for insulin-regulated glucose-4 translocation and glucose transport. And that chromium picolinate improves insulin action by protecting against the pathological increase in membrane cholesterol that compromises F-actin, which is a requirement for proper insulin-regulated glucose transport for translocation and glucose transport, but it's not something that was affecting necessarily the insulin signaling itself. Okay, all right. Any questions about this before we head out? Doubts? All right, everyone. So if you have any questions with regards to the draft, you can send me an email and I can help you with anything. Okay. All right. Have a great weekend.
Thank you.